My name is Stephen Eschrich, and I'm from the Moffitt Cancer Center. The title of my poster is Artificial Intelligence-Based TMT Experiment Planning Tool. As most of us know, tandem mass tag, or TMT proteomics, is a barcoding approach for multiplexing samples in a single mass spec proteomics experiment. Our group recently published a paper using 29 TMT 6-plex experiments in a large-scale analysis of lung squamous tumors. From this experience, we have identified factors that impact proteomics and can be significant in large-scale projects. So the question that we asked ourselves was, how can we help ourselves and others design new experiments taking these things that we've learned into account? The things that we've learned are often called heuristics in the AI research community. Heuristics are rules of thumb when using a specific technology or things that have come generally from experience. Some examples in proteomics include sample randomization or distributing replicates across experimental batches. Another area of AI research is constraint satisfaction problems. This research originated in the 1970s in AI development. Some examples of constraint satisfaction problems include Sudoku puzzles, the 8 queens problem, and school schedules. As an example of school schedules, we need to find a school schedule that works for teachers, students, and classrooms with constraints such that teachers and students can't be in 2 places at one time. Constraint solvers are techniques that find solutions to problems given a set of limitations or constraints. So therefore, experimental design can be considered a constraint satisfaction problem. We arrange samples or replicates on TMT plexes subject to these constraints. We ensure calibration channel is always in the last channel, for instance, for consistency and to avoid bleed overs in signal, balancing conditions across TMT plexes, and balancing replicates across TMT plexes, as well as maybe not placing replicates next to each other, spread them out over the TMT experiment. Optiplanner is an AI-based constraint solver. It's an open source tool for planning and scheduling. Within the system, the user can define constraints, which are limits to the searching in the solution space. There are 2 types of constraints: hard constraints that cannot be violated, and soft constraints, which assign numerical penalties to specific conditions. Therefore, we developed the Tmt Planner. It is a web-based tool for designing experiments based on heuristics of TMT experimental design. A user needs to define a sample set, the number of TMT experiments, and the phenotypic variables. Tmt Planner identifies a solution or experimental layout that meets the constraints imposed by the heuristics defined. A graphical experiment design tool will allow the user to override the suggested layout. After the planning is completed, a worksheet can be generated with TMT labeling assignments and other experimental parameters for further use. This is an example of the main screen for the TMT Planner. The first step in designing an experiment is to determine the number of samples, the number of conditions, and the channels. The channels would be the TMT 6-plex, 10-plex, 11-plex, or 16-plex. The user can also alternately upload an experimental design within a CSV or Excel file. The format for the experimental template is available in the tool. The second step for the experiment design is to set the options for the experiment. These include having a calibration channel, balancing conditions across plexes, and other key characteristics. The third step of the process is to find the number of replicates for each biological sample. Here we've chosen to have 5 samples, 2 conditions, and a TMT 11-plex. We would like to have 2 replicates for each biological sample. I'd like to point out in the upper right-hand corner the auto padding option. Often in a TMT experiment, there may be too few samples given the number of TMT labels that are available. The software will automatically assign a new replicate, 1 replicate per biological sample in round-robin fashion until the total number of TMT labels have been used. The fourth step is to enter condition values for each sample. Note that there are more replicates per sample than we originally had provided. This is due to the padding, auto-padding approach as previously described. Also note the red button in the upper right-hand corner. This indicates an autofill of sample conditions based on the first replicate. For instance, if we put drug treated as the first condition for the first sample, this actually will be autofilled into all subsequent replicates of the same sample. Once all of the conditions have been entered, you will note that the sample names will be transformed to include the actual conditions within the sample name for clarity of sampling labeling. The final step is to design the experiment. This is where the Optiplanner tool is used to find a solution satisfying the heuristics previously defined. The way that we approach this is to allow the Optiplanner optimization to run for 10 seconds and provide the best layout found. On the left, you can see the proposed layout. There are two TMT11 plexes with unique identifiers, and you can see samples associated with each of the individual labels. The tool allows for the samples to be dragged and dropped wherever a user may want after the initial experiment design has been provided. A close-up, you can see that some things to notice are the order is random, the replicates of the same sample are not adjacent, conditions are split across TMT experiments, and the calibration channel is last. I'd like to acknowledge several of my collaborators, including John Kuman and Eric Hara. I'd also like to acknowledge Scott Kukris, who did the software development for this tool, as well as the Moffitt internal funding for this project. In conclusion, this is an evolving tool based on user feedback. Feel free to contact me at the email address if you'd like to discuss further. Thank you for your time and attention.